everyone. This is Dr. Anita Srivastav, and today I am going to talk about industrial microorganisms, another lecture of our industrial microbiology course. So, industrial microorganisms, they are actually known as the backbone of any industrial microbiology process, and they are grown on a large scale to form products or they carry out chemical transformations. So, microorganisms are very important in any fermentation process or any microbial process. So, before going into the depth of or the detail of the microorganisms, I would like to brief about the history and the background knowledge about the microorganisms. Microorganisms actually scientifically they were exploited by the study of Louis Pasteur in the 19th century, and he explained the role of microorganisms in the product formation, and they played an important role in the formation or the production of food and beverages. But talking about Louis Pasteur, it was Anthony von Leeuwenhoek 200 years before the studies of Louis Pasteur. He was the first scientist to tell about the existence of living organisms or these microorganisms under microscope by his own made lenses. And even history reveals many applications of microorganisms in the production of food and beverages. But talking about the alcohol production, this alcohol production is around 6,000 BC back. But the alcohol formation was known to occur in the presence of or by the involvement of some chemical processes. At that time, no one knew about the microorganisms or the role of microorganisms, which kind of microorganisms playing the role in the alcohol formation. So this is the story actually, that how does these microorganisms came into existence and about their knowledge. So the most important characteristics feature of these microorganisms that they have the capacity to convert nutrient or substrate into an end product. And talking about industrial microbiology. Industrial microbiology, it was revolutionized and sparked with the discovery of some antibiotics. Among them, the first antibiotic that was discovered was penicillin by Penicillium notatum in 1940s. Then, with the advancement in genetics, in microbial genetics, that was the genetic manipulation of microorganisms, and this technology was known as the recombinant DNA technology. So the development of this technology actually took this industrial microbiology to a different level, as these recombinants they are used for the production of so many products which are very much useful and beneficial for the humans and for the mankind, and they are useful in the treatment of certain diseases, so they are very important. And I will talk about this recombinant DNA technology in my next lecture, when I'll be talking on the strain improvement of these industrial microorganisms. So this technology will be talked in detail. Now, going to the key properties of microbial processes. There are three key properties that are involved in any microbial processes. First one, microorganisms, medium, then product. So microorganisms, when we talk about any industrial process, any microbial process, so it is very important to know the features of the microorganisms, whether it has the potential to be used in industry or not. So certain characteristics or certain qualities that microorganisms should have before being used in any industrial process is they should have a potential to be useful in the formation and the production for the products that is for human use, that is for the treatment of certain diseases in food supplements, etc. The microorganisms, they should be able to produce enzymes and proteins which will be used in different industries. There are so many enzymes, microbial enzymes, that play a very important role in some industries, like they are widely used in textile industries, in agriculture, in food, in pharma. So they have a very important role when we talk from the industrial aspects. And proteins are also very important. Amino acids, we are talking about the proteins, we are talking about the amino acids. Then the microorganisms, they should have the capability to grow very fast. That means they should have a low doubling time. Low doubling time means generation time should be less. Means when we will talk from the fermentation point of view or for many industrial processes, the run should be shortened. It should have a the any process any it should have a short run. Then obviously these microorganisms, they should be non-pathogenic. They should not cause any pathogenesis. So this is about the features of the microorganisms. Then talking about the medium. So first of all, as previously also I discussed, that media formulation is a very important aspect in any industrial process. So the medium which is used, it should contain the substrate that is highly economical and easily available in large quantities, as in industries it is a scale up process. It is being used on large scale, so large availability or means easy availability in large quantity of the substrate is very important. Then all the waste products like from agriculture, dairy and paper industries which have nutrients, which have nutrients, so they should be utilized for the formation of useful product by using these nutrients, which are actually the waste product from some industries. Talking about the product, so as we know that industrial fermentations, they are used on large scale containing the microbial cells and unused constituents of the medium, and an efficient and economical method. So basically after the scale up or after the upstream process comes the downstream process. Means we are talking about the recovery and purification parts. So it is well known that almost 60 to 70% of the process costs for the recovery and the purification. So an efficient and economical method is exploited and it must be developed to obtain the desired products and with a minimum loss. So this is related to the product. Now talking about the major commercial products of microorganisms. So the major commercial products of microorganisms are microbial cells, microbial enzymes, then we have primary metabolic products, secondary metabolic products. So metabolic products, before understanding the primary and secondary metabolic products, we should understand that what are metabolites. So these metabolites, they are the intermediates of products during metabolism. And a a primary metabolite, it is directly involved in the normal growth, development, and reproduction. And are the examples of primary metabolites are ethanol and lactic acid production. Then talking about the secondary metabolic products, first thing, secondary metabolic products, they are not essential for cell growth. They are not directly involved in the processes, and examples are antibiotics. These secondary metabolites, they are low molecular mass. They are low molecular mass molecules which are not essential for the growth and survival of the producing microorganisms. They are synthesized in the late phase of growth cycle, that is in the stationary phase when we talk about the growth curve of any microorganisms. So these secondary metabolites, they are actually produced during the stationary phase, whereas primary metabolites, as they are the growth, they are essential for the growth, they are produced during the growth phase. So then third, then this one, the recombinant products formed by DNA recombinant technology. Means, as I was talking about the recombinant technology, which will be discussed in detail in the next lecture. And chemicals that are produced by the microbes by using medium components or the substrate. Now talking about the industrial products. So as the development of the industrial microbiology sparked from the discovery of the antibiotic. So antibiotics, they have a major role in industrial microbiology, and along with antibiotics there are other products which have also a major impact, and these are beverages. As beverages, they are being used since like in BCs, then organic acids, enzymes, vitamins, single cell protein, and specialty products. So we will be discussing all these industrial products in detail. Now talking about some microbial products and the processes that are of great interest in industrial microbiology, means the involvement or some known microorganisms which plays an important role in industrial microbiology. So talking about the industrial products, Saccharomyces cerevisiae plays an important role in the production of ethanol, where glucose is used as a substrate, and whereas Saccharomyces fragilis is also exploited, but it is also producing ethanol, but it uses the substrate lactose. Then another example is Clostridium acetobutylicum, means product is acetonol butanol. Butanediol is produced by Enterobacter. Then talking about the enzymes, about the production of the microbial enzymes, that so a number of strains, for example, Aspergillus, Bacillus, Mucor, Trichoderma, they are widely used in industries in microbiology for the microbiology purposes. Then agricultural products, gibberellins by Gibberella, and then coming to the food additives, Corynebacterium is used for amino acids, Aspergillus niger for the organic acid, for example, citric acid. Then we have nucleotides, vitamins, so different microorganisms are used for different products. And then talking about the medical products, we know antibiotics, it is related to the penicillium, streptomyces, bacillus. and insulin or the human growth, that is E. coli, and then finally for biofuels, if we talk about biofuels and in the production of the hydrogen, it is photosynthetic microorganisms. For methane, this is methanobacterium, and for ethanol, this is zymomonas thermoanaerobact. Now coming to the antibiotics. So as we all know that when it comes to the name of antibiotic, we should remember Alexander Fleming, because he was the he was the scientist who was known to discover penicillin. So Fleming and penicillium, these both are associated. In antibiotics, talking about the antibiotics, they are chemical substances produced by certain microorganisms. How does they function? They function either by killing or inhibiting the growth of harmful microbes without affecting the host cells. And it was first, penicillium was the first antibiotic discovered by Alexander Fleming in the year 1929 from the mold Penicillium notatum. And there are many other antibiotics produced by microorganisms, and they include streptomycin and other antibiotics used to treat a number of bacterial infections. So now talking about the mode of action of antibiotics. Antibiotics, the role they play are they inhibit the cell wall synthesis of the bacteria, they damage the cytoplasmic membrane, and inhibition of nucleic acid and protein synthesis. They also inhibit the specific enzyme systems. So these are some basically the mode of action of the antibiotics which I have told in brief. Now some common antibiotic producing microorganisms. So we should know about the names of some microorganisms which are involved in antibiotic production. And this is when we talk about the bacitracin, the antibiotic, Bacillus subtilis plays an important role. Bacillus polymixa for polymyxin. And talking about the actinomycetes, Streptomyces nodosus rhesus amphotericin B, Streptomyces venezuelae, chloramphenicol, Streptomyces aureofaciens produces chlorotetracycline and tetracycline, Streptomyces erythraeus, erythromycin. So there are several other microorganisms producing antibiotics. Talking about the fungi, very well known, we know about the cephalosporin, cephalonin, then penicillium grevisiofilum producing greciofilvin, then penicillium notatum for the production of the antibiotic, that is the penicillin. Now when we come to the beverages, actually, so talking about the beverages, we know that yeast, they are widely used microorganisms for the production of the beverages like beer, brandy, rum, wine, whiskey, etc. And this is the process that is actually taking place. That yeast, in presence of using the substrate carbon source as the sugar, they produce alcohol and carbon dioxide and is released fermentation, and it is basically an anaerobic process. Then in industrial processes, Saccharomyces cerevisiae, this is also called as brewer's yeast, and it is used in fermenting fruit juices and malted cereals to produce ethanol. And once after the fermentation, these beverages, they are distilled to produce both alcoholic and non-alcoholic beverages, that is including whiskey, brandy, rum, etc. And this is about that actually the this is the production of the ethanol. That is when we talk about the in during the carbohydrate metabolism. In the previous lecture, I talked about anaerobic fermentation, which ended with the product like ethanol production and the lactic acid production. So here, the glucose, which is used and which is the substrate for the glycolytic pathways. So glycerol and through the glycolysis with the generation of the 2 ATP, it formed pyruvate. Then pyruvate, after the decarboxylation, that is the removal of two carbon dioxide molecules, it formed two molecules of acetaldehyde resulting in two ethanol molecules. And here NADH is being converted to NADP in order to replenish the NAD which is required during the glycolysis. So this reaction ended with the production of ethanol from the glucose in under anaerobic condition, anaerobic fermentation. Then talking about the organic acids, microbes, they are also used in the industrial production of certain organic acids. Like citric acid, it was first discovered organic acid from the microbial fermentation of a lemon, that is citrus fruits, and citric acid was the first organic acid to be introduced. Organic acids, they are also produced directly from the glucose, and here we can see the formation of the lactic acid from glucose. So glucose under glycolytic pathway, it is producing glyceraldehyde 3 phosphate. Then in the presence of glyceraldehyde 3 phosphate dehydrogenase 1,3 biphosphoglycerate is formed, and this 1,3 biphosphoglycerate forms pyruvate. This is during the glycolytic pathway. This is Embden-Meyerhof pathway, EMP pathway. So now this pyruvate which is formed in the presence of lactate dehydrogenase, it forms lactic acid, and one NADH is converted to NADH plus H ions. So here in case of the formation of the 1,3 biphosphoglycerate from the glyceraldehyde 3 phosphate, one NAD was resulted in the formation of NADH plus H ions. So basically the purpose of this lactic acid fermentation just to replenish this NAD for the formation or for to help the process of the glycolytic pathways. Now talking about the products of the organic acids, there are certain organic acids which are known: acetic acid, citric acid, fumaric acid, gluconic acid, itaconic, kozic, lactic acid, and there are different microorganisms which are used and the industries where they are useful. So acetic acid is produced by the acidobacters and they're widely used means as a variety of food uses. Citric acid is used in pharmaceuticals and as a food additives. Aspergillus niger is used in molasses, that is a molasses based medium. Fumaric acid is from Rhizopus nigricans in the manufacture of resin, tannin, and casein. Gluconic acid by Aspergillus niger in glucose mineral salt solutions, and it is basically a carrier for calcium and sodium. Then we have itaconic acid by Aspergillus terreus, esters which can be polymerized to make plastics. So this way we can say, and one more important, lactic acid also. This is a homofermentive Lactobacillus delbrueckii. It is as a carrier for calcium and as an acidifier. Now coming to the enzymes. So we all know that enzymes are biological catalysts. They control the biochemical reactions in the living system. They are widely used range of applications in the production of both medical and non-medical field. They are equally important in both the fields. And apart from the plants and animals, enzymes, they are also obtained from microbes. Here we are talking about the microorganisms, so we are interested in the enzymes which are formed from the microbes, and therefore they are also called as the microbial enzymes. And there are different types of enzymes which are known, like amylase, protease, lipase, pectinase, cellulase, xylanase, chitinase, sucrase, and phosphatase. And these microorganisms, they are basically or majorly used for the production of industrial enzymes through the safe gene transfer methods. And the first industrial produced microbial enzymes, it was obtained from the fungal amylase, and long before in year 1896. and it was used to cure indigestion and several other digestive disorders. So basically, if we look to these different type of enzymes, here we can see the substrate which they are utilizing and the microorganisms which were widely used for the production of these enzymes. So they includes the aspergillus, saccharomyces, bacillus, and streptomyces, and different other microorganisms. Now talking about the vitamins. Vitamins, they are the organic compounds which are capable of performing many life-sustaining functions inside a body, and they are actually essential micronutrients which are required in small quantities for the body's metabolism. Vitamins are not synthesized in a body, but they are supplied through the diet. But some microorganisms, they have the potential to synthesize vitamins along with animals and plants. And inside the gut microflora, the microorganisms, they are capable of synthesizing vitamins. And these microbes, they are involved in synthesizing vitamin K, vitamin C, beta carotene, biotin, aldosterol, folic acid, vitamin B12, thiamine, pantothenic acid, riboflavin, and pyridoxine. And the microorganisms which are involved in vitamin synthesis, they are Pseudomonas denitrificans, Bacillus megatherium, Streptomyces olivaceus. They are some vitamin B12 producing microorganisms. When talking about the beta carotene provitamin, Blackislia trispora, Phycomyces, and this Blackislians, they are used for producing beta carotene and provitamin. Talking about the single cell protein. Single cell proteins, they are microbial cells which are rich in protein content, and they can be used as protein supplements for human and animals. And spirulina. Microbes like spirulina, they can be grown easily on materials like wastewater from potato processing plants, which contains starch, high content of starch, and others are straw, molasses, animal manure, and even sewage. They produce large quantities and they can serve as food rich in proteins, minerals, fats, carbohydrate, and vitamins. So single cell proteins, they are also a product which can be synthesized by different microorganisms. Now moving to the specialty compounds that is related to the medical and health which are produced by microorganisms. So we'll talk about the polyethers, enzyme inhibitors, bioherbicides, anabolic agents, anti-tumor agents. So there's number of microorganisms or the sources which are responsible for the production of these compounds, and these are all the detail about the specific products which are being produced and the process which they are undergoing and the treatment which they are going for. So we can see here a different strains like streptomyces, some and aspergillus, penicillium, and this is all streptomyces actually. So and claviceps. So these are some strains, they are being used for the production of some specialty compounds which are widely used in medical and health. So that is all for the industrial microorganisms, and in the next lecture I am going to talk about the strain improvement of the industrial microorganisms. Thank you.